All right, thank you. Uh, Thanks so much. And I will present our recent work on the quantum chemical investigation to the volatile diffusion and also their vibrational spectra, especially in irradiated lunar samples. So first of all, a very brief introduction. So in 2017, Schreider's paper analyzed the MQ data, especially for the 2.8 micrometer features, which is an indication of hydroxyl or water molecules. And then later in 2019, Manuel Hendricks used the data from RO and also Divina radiometers to probe the temporal changes of those water. And they actually found a very clear diurnal variation. So the local water abundance varies with the local time and also temperature. So essentially, this points to the idea of very active volatile cycle, starting from solvent implantations where hydrogen combined with oxygen in the regulars, and also the thermal desorption allowing the volatiles to release from the surface. And also the diffusion, which is kind of in between, allows the volatiles to migrate among the regulars and also release. All right, so recently there was a paper by Liam Morisi, who used reactive MD simulation to understand the diffusion of hydrogen in this amorphous silica. So one of the key findings is that in a perfect, pristine silica surface, the hydrogen diffusion activation energy is too low to explain the diurnal variation. But if you consider those irradiation-induced defects, those defects is going to effectively trap those hydrogen atoms, and also there could be further chemistry going on, like water formation. So the takeaway here is that especially the damaged state of the lunar regulars is a critical point if you're doing the volatile modeling, especially for the diffusion process. All right, here, so in this study, we use a combined models to understand the diffusion, especially with those defect sites. And we choose three key volatile species like atomic hydrogen, molecular hydrogen, and also molecular water. So the question we want to ask is how exactly those defect is trapping those hydrogen-bearing species, and also what kind of binding it is between those defect site and the volatile species. So here we use a combined simulation framework. First of all, the molecular dynamic simulations, including the ab initio MD and also reactive MD. So essentially, this is to model the diffusion process and also the dynamic process of breaking of H2 and H2O, possibly. And also the quantum chemistry by density functional theory to calculate the high resolution IR spectra for some particular structure. So for the damaged regulars, we have this point defect, including removing one silicon items from this pristine silica unit cell, and also oxygen vacancy by removing one oxygen item. We also have this large-scale damage surface, which is like similar to vesicle, which are largely found in lunar regulars. All right, so starting from the diffusion of H2 and H2O, so this is similar to the scenario that you have H2 and H2O diffusing among the regulars, and then you meet a defect site created by irradiation. Or alternatively, this can also happen if you have H2 and H2O trapped interstitially in the silica regulars, and a slowing irradiation is going to create a defect nearby. So the result for this H2 and H2O diffusion is that the defect energy is very, very high, and it can effectively trap those H2 and H2O and also dissociate them into hydroxyl groups. Sorry. So this is actually not surprising because we have seen a lot of this hydroxylation process if you're doing this silica water interface. But this actually simplifies the problem because the diffusion of H2 and H2O has been reduced to a province of hydrogen diffusion or hydroxyl diffusion. So this hydrogen diffusion, the first thing we identify is this hydrogen actually jumps through this 4 oxygen vacancy sites, oxygen dangling bonds, and also forming those temporary hydroxyl groups. But the hopping is very, very fast within the time scale of 20 femtoseconds. So essentially, this 20 femtosecond is very close to the hydroxyl stretching timescale. And as a reference, the hydroxyl stretching vibration timescale is, if you consider 2.8 micrometers, the timescale is 9.3. And also the overall lifetime of this hydroxyl stretching is about 1 picosecond before the excited hydrogen loses their energy. So essentially what this means is the hydrogen is going to only complete like 2 cycles of stretching before they hop to another oxygen site. And this will effectively affect the 2.8 stretching mode and possibly like broaden the spectrum and also lower the intensity. And with the longer simulation, we actually identify a relatively more stable structure. This hydrogen is going to bridge between 2 dangling oxygen atoms. And this is not unique for this point defect simulation, but also this has been found largely in those vesicle simulation as well, like H2 and also H2O trapped in this vesicle. So this is a kind of interesting question because obviously this hydrogen is not going to form 2 covalent bonds with 2 oxygen atoms, but it is a hydrogen bond or it is just a hydrogen hopping between those 2 oxygens. So to answer this question, we run a longer AMD simulations, and the answer is actually yes to both because we found based on the charge density distribution, and we found there's a covalent bond exist for this hydrogen between 1 oxygen, and there's also like a charge exchange between this hydrogen to another oxygen. So both the hopping and also the hydrogen bond exist. And also this hopping is very, very fast. And simply because the hydrogen is very close between those 2 oxygen sites, and the hydrogen bond actually facilitates this diffusion process. And the time scale is about like 0.1 picosecond, which is relatively longer compared to hopping between those 4 oxygen sites, but still like very, very short compared to the lifetime of hydroxyl stretch. And we also did a DFT calculations of the vibrational analysis. So this is done by systematically perturb the items from its equilibrium positions, and then we calculate the Hessian matrix and also the overall energy surface. So what we found is actually a redshift to this 2.8 micrometers. Thank you. And this is just because the hydrogen bond is actually weakening the covalent bonds of their original host oxygen atoms. And the intensity is a little bit larger because, again, this is because the hydrogen bonds and make the dipole moment larger, and also the hopping happens even faster. The silicon oxygen stretching is now affected by a lot. It's relatively more stable. But again, this feature is now very stable because, again, we have shown in the previous slide that the diffusion actually happens in 0.1 picosecond, and essentially this is only like 10 cycles of hydroxyl stretching, and it's still not a complete like 1 picosecond stretching lifetime. So here, the takeaway here is that we have simulated the defect-induced diffusion process, and the defect is strong enough to trap all those H2 atomic hydrogen and also molecular water. But the way that the trapped hydrogen is actually forming this bridging between 2 oxygen dangling bonds. And essentially, we have also performed this vibrational analysis to show that this trapping is not necessarily give you a 2.8 micrometer features because of the complicated hopping and also the stretching. And the main takeaway here is we definitely need high resolution error spectral observations to fully understand the volatile cycle on the lunar surface. Thank you.